Okay, I'm going to go ahead and start the media review for Microbiology Exam 3. This will be for the lecture and for the lab. I'll start with the lab portion. There are a few photographs that I'm going to show you. Now, for the purposes of the lab, just remember, the pictures that you're going to see are not so essential. I mean, from the information alone that I give you in the question, you should be able to give the response. So it's not so much recognition, though it does help if you can recognize the organisms. Then in many cases, it may give you the answer right away. So the first picture that you're looking at is for Salmonella. If you remember, Salmonella has multiple flagella that you can see on the organism. I would like for you to know, and remember this exam will cover Lab Exam 6. Have a question already? Yeah, it's not. Oh, it's not showing. Oh, that would be important, wouldn't it? So you can't actually see Salmonella the way I can, huh? Ah, there it is. Yes? Okay. So remember, this comes from your lab assignment number 6. You should be able to list all of the enteric organisms that are related to gastroenteritis. So first portion of the exam, first portion of that presentation. Has everybody been able to access the presentation? Yeah, for lab assignment 6? Am I getting nodding of the head? Yeah, okay, all right. So, so you should be able to list all the microorganisms referred to as enterics that are related to gastroenteritis. You should know the hallmark signs and symptoms of this salmonella infection. Remember, it causes typhoid fever, so you should know the cardinal signs and symptoms or characteristics of typhoid fever. You should know what the typical treatment for salmonella is, for salmonella typhi. The characteristics of salmonella gastroenteritis, that's also known as food poisoning, has some typical characteristics, and you should be familiar with that as well. Now, another organism that you should be familiar with, and that's also a part of that lab assignment, is Shigella. Shigella. Shigella is commonly associated with a specific disease process. You should be familiar with that disease process. You should know what the characteristic signs and symptoms are of a Shigella infection. Dysentery syndrome, is related to Shigella. It's due to virulence factors, and you should be familiar with the virulence factors that are associated with the Shigella infection. Another organism you should be familiar with is Vibrio cholera. Vibrio cholera. You need to know the general characteristics of Vibrio cholera. Again, you can see this is a flagellum, unipolar flagellum on the organism, easily recognizable. But you should be able to describe the general characteristics of Vibrio cholera. Another organism you should be familiar with is Campylobacter. Campylobacter, another of the enteric organisms. Should be able to identify the general characteristics related to Campylobacter. And then finally, you should be able to recognize Helicobacter pylori as the organism. What I'd like for you to be able to tell me is the particular type of environment that Helicobacter pylori can exist in, where specifically in the GI tract you would see Helicobacter pylori, and then what type of diagnostic studies can you do to diagnose Helicobacter pylori infections. And we'll go with the lecture portion of the exam. You should be able to define and describe non-specific resistance. What is meant by the term non-specific resistance? You should be able to tell me what the ciliary escalator is. ciliary escalator is a type of non-specific resistance. You should be able to tell me where it's located at, what's involved in it. Should be able to tell me where lysozymes are found at. Where are lysozymes found at? How are lysozymes a part of this non-specific resistance? Bacteria have two types of enzymes, or at least some bacteria have two types of enzymes that help protect the bacteria. These are catalase and superoxide dismutase. You need to explain those two enzymes to me, how they're important in protecting bacteria. You need to know about inflammation. You need to know about inflammation cold. You need to tell me what the characteristics of inflammation are, why inflammation is important. Should certainly be able to tell me the physical characteristics that you see in inflammation. Should be able to give me the steps of inflammation. What do you see in the inflammatory process starting at step 1? About 6 steps involved in inflammation. You should be able to go through each of those steps. You should know what normal microbiota refers to. The other term that's used is normal flora. You should be able to tell me how normal flora can provide protection against different types of infection. You should know the importance of eosinophils. Eosinophils. Eosinophils are very common in a particular type of infection. The term sometimes is used, and I'm not sure if your book used this term, but you can look this up in the index. Eosinophilia. Eosinophilia is very common in a particular type of infection. You need to be familiar with eosinophils and eosinophilia. You need to know the term margination, and what that refers to. I'm looking at the third question as it relates to inflammation, the function of inflammation. So there's at least 3 questions about inflammation on the exam. The symptoms of tetanus are related to a specific structure or property of the Clostridium bacteria. I need to know what the symptoms of tetanus are related to. I need to know how most pathogens gain access through the integumentary system. How do most bacteria gain access through the integumentary system? I might have jumped chapters on you with this, but if I'm putting it on the review, it's fair game. The polio virus. How does the polio virus gain entry into our system? How does it gain access into the system? What's its portal of entry? Which means obviously you need to know what the term portal of entry means. You need to know what a superantigen is, a superantigen. How a superantigen relates to septic shock in gram-positive organisms. Superantigen, how it relates to septic shock in gram-positive organisms. This will make you read at least part of the chapter. I need to know what the most common portal of entry of pathogens into the human body is. What's the most common portal of entry? What's meant by the term parental root? Parental root. Need some examples of parental roots. When an organism enters the body via parental route, need some examples of that as well. You should be familiar with the signs and symptoms of helminth diseases. You should be familiar with the signs and symptoms of protozoan diseases. Where do we see what causes those symptoms? What part of those bacteria cause specific symptoms to occur? You should be familiar with the term endotoxin and the term exotoxin. What type of organism would you see endotoxins in? What type of organisms would you primarily see exotoxins in? Where in the cell would you see exotoxins and endotoxins? When I say the cell, I mean the bacterial cell. In gram-negative organisms, sometimes when we treat these organisms with antibiotic therapy, we can sometimes make the patients worse by precipitating a condition called septic shock. I need to know how that mechanism would take place. I'll repeat that one more time. Individuals that have gram-negative infections can be treated with antibiotic therapy, but in treating the person that's infected by that organism with antibiotic therapy, sometimes we can make the person worse. Explain how that mechanism happens. How can botulism be prevented? How can botulism be prevented? I mean, other than not getting in contact with botulism. Another question about nonspecific resistance. I need to know the chemical factors that help protect the skin and the mucous membrane from infection. Chemical factors that help protect the skin, the integumentary system, and the mucous membrane from infection. I need to know a definition of the complement system. I need to know the chemical agents inside the body that can lead to vasodilatation. You can find that around the inflammatory response. Chemicals in the body that can lead to vasodilatation. I need to know what's meant by the classical pathway of the complement system. What activates the classical pathway for complements? There's another question about inflammation, describing the characteristics of inflammation. Might have jumped chapters on this one as well, but if I put it on the review, it's fair game. I need to know what's meant by complement fixation. Complement fixation. Actually, I think that is described in chapter 16. I may be wrong. That might be a chapter 17 question. But again, it's on the review now, so it's fair game. It's only one question. I need to know what the effects of complement activation are. What are some of the effects of complement activation? Need to be able to define the term opsonization. Opsonization. You should be able to tell me the effects of histamines. What cells release histamines? What are the effects of histamines? You should be able to define a chill. What's a chill? What is a chill reflective of? Chill. C-H-I-L-L. Chill. The big chill. Okay, that's it. If you know those things, you should do fine on the exam.